Hello everyone and welcome back to Bioresource, Technical Resources in Biotechnology. Today we are diving deep into the fascinating world of chemiluminescent immunoassay, CLIA, a highly sensitive and widely used diagnostic technique in clinical and research laboratories. This session will help you understand the principles, techniques and the applications of CLIA. So let's get started. Before we dive into CLIA, let's understand what an immunoassay is. Immunoassays are biochemical tests that detect or quantify biomolecules using antigen-antibody interactions. They play a crucial role in clinical diagnostics by detecting minute concentration of biomolecules such as hormones, tumor markers, infectious agents, and therapeutic drugs. Some key areas of applications include hormonal testing, tumor markers detection, infectious disease detection, therapeutic drug monitoring, and autoimmune disorder detection. Over the time, immunoassay techniques have evolved to improve sensitivity, specificity, and safety. Starting from the radioimmunoassay to chemiluminescent immunoassay. Radioimmunoassay is the first immunoassay technique using radioactively labeled antibodies. While highly sensitive, it poses radiation hazards. The use of enzyme conjugated antibodies and colorimetric reaction for detection called the ELISA, which is safer than radioimmunoassay but has a lower sensitivity than chemiluminescent methods. Chemiluminescent immunoassay uses chemiluminescent label that emit light on reaction detected by a luminometer. CLIA provides higher sensitivity, a wider dynamic range, and faster reaction times than the ELISA. So the CLIA provides higher sensitivity, faster reaction times, and a wider dynamic range compared to the traditional immunoassays. So what is chemiluminescence? Chemiluminescence is a phenomena where light is produced through a chemical reaction without requiring external light excitation. Unlike fluorescence, which requires an external light source, chemiluminescence is a self-sustained through oxidation reactions. This property makes it ideal for diagnostics as it eliminates background noise and enhances signal clarity. The examples include the luminol reaction, commonly used in forensic science, is a classic example of chemiluminescence. Another important system is the acridinium esters, which react with alkaline hydrogen peroxide to produce bright luminescent signal. The energy released during the reaction is converted into visible light instead of heat, which differentiates it from fluorescence or phosphorescence. As you can see, the different features of chemiluminescence and fluorescence. In simple terms, fluorescence requires external light source while chemiluminescent is self-sustained via chemical reactions. Let's look at the role of catalysts or enzyme substrate reactions in the light emission. Chemiluminescence occurs due to oxidation reactions which often requires catalysts enhance efficiency. In CLIA, the light is generated by enzyme catalyzed reaction involving specific substrates and label. So first one is the luminol reaction. It's a classical CLIA example. Luminol undergoes oxidation in presence of hydrogen peroxide and a catalyst, horseradish peroxidase or the HRP. The reaction produces an excited state intermediate which emits light upon returning to the ground state. Second one is the acridinium ester based chemiluminescence. The acridinium esters react with alkaline hydrogen peroxide to generate light. It is common in automated CLIA platforms due to high sensitivity and stability. Next is the enzyme-based chemiluminescence, which involves the HRP and ALP systems. HRP, the horseradish peroxidase, catalyzes the oxidation of luminol in the presence of hydrogen peroxide, leading to photon emission. ALP, also known as the alkaline phosphatase, reacts with chemiluminescent substrates like AMPPD to emit light. These enzyme reactions ensure highly sensitive detection of biomolecules in clinical diagnostics, pharmaceutical research, and food safety. So in CLIA, the chemiluminescent reaction provide high sensitivity, stability, and rapid result, making it ideal choice for diagnostic applications. Now that we understand chemiluminescence, let's discuss how it is applied in immunoassay. CLIA works on antigen-antibody interactions where a chemiluminescent label is attached to either antigen or antibody. When a trigger solution is added, the label undergoes chemical reactions emitting light proportional to the unlight concentration. The emitted light is detected by a luminometer which quantifies the unlight based on the standard curve. So here's one example. In hormone testing, such as detecting TSH or the thyroid stimulating hormone, CLIA can measure very low concentration of the hormone with high accuracy. So let's get into the details. First one is the antigen antibody binding in CLIA. The chemiluminescent immunoassay relies on the specific interaction between an antigen and an antibody, forming this basis for detection. The binding can occur in several formats. One is the direct binding, sandwich assay, competitive binding, etc. This step ensures selectivity, making CLIA a highly specific and sensitive in detecting the target molecules. Second step is the chemiluminescent reaction for detection. The detection mechanism in CLIA involves chemical reaction that produces light emission. The key steps include a chemiluminescent label such as acridinium ester, luminol, or enzyme-based system like HRP is attached to an antigen or an antibody. When a specific trigger solution, the oxidizing agent, is added, the label undergoes a chemical reaction producing an excited state intermediate. This intermediate releases energy in the form of visible light as it returns to its ground state. The intensity of the emitted light is directly proportional to the amount of sunlight present in the sample. Higher the antigen concentration lead to stronger luminescence, allowing for the quantitative measurement. This principle ensures that CLIA provides high dynamic range and sensitive detection even for very low concentration of analytes. For measuring the signal, a luminometer is used to detect and measure the emitted photons. The instrument contains a photomultiplier tube or a CCD sensor to capture the light intensity. The data is then processed and analyzed, converting luminescence intensity into unlight concentration using a standard curve. CLIA surpasses ELISA in sensitivity and speed, and it is widely used in hormone assays, infectious disease diagnostics, and tumor marker detection. And the method provides automated high throughput capabilities in modern clinical laboratories. So the takeaway is that the CLIA's detection mechanism allow high sensitive quantitative measurement of the biomolecules. Chemiluminescent immunoassays is classified into different types based on the labeling strategy or the binding mechanism used for detecting an analyte. Each type has unique advantages and applications. So let's look at each one of these in detail. In direct CLIA, the chemiluminescent label is directly attached to the antigen or antibody. The light signal is generated directly proportional to the analyte concentration. The advantage is that it is simpler assay design, meaning that only fewer steps are involved, faster reaction time due to direct detection. The main drawback is that lower signal intensity compared to the indirect methods, a limited signal amplification which can reduce the sensitivity for the low abundance analytes. One of the example is the use of this assay in hormone assays like TSH and insulin, where high specificity is required. Coming to the indirect CLIA, a secondary antibody is labeled with the chemiluminescent marker is used to detect the primary antibody bound to the analyte. So the advantage is that the signal gets amplified and which increases the sensitivity. Higher signal intensity due to multiple secondary antibody binding sites is one of the key advantage and increased sensitivity compared to the direct CLIA. But the problem is that it requires additional incubation and washing steps that increases the assay time. Example include autoimmune disease diagnostics such as detecting anti-nuclear antibodies in lupus testing. Next is the competitive CLIA. The analyte in the sample competes with a labeled analyte for antibody binding sites. The signal intensity is inversely proportional to the analyte concentration in the sample. The advantage here is that it is suitable for small molecules and haptens that cannot be captured in sandwich format, and it is useful for drug monitoring and toxic detection. The major disadvantage is that the more complex assay design is required due to competitive binding, and it requires careful standardization to ensure the accurate quantification. Example of this is the therapeutic drug monitoring. Next is the sandwich CLIA. The sandwich CLIA uses two specific antibodies. One is the capture antibody immobilized on a solid surface, and a detection antibody labeled with a chemiluminescent marker. And this format ensures higher specificity and sensitivity. Major advantage is that it is highly specific. It is best for detecting large proteins and biomarkers and minimal interference from other molecules. But the drawback is that it requires high quality antibodies to prevent the cross reactivity, and it is not suitable for small molecules that cannot bind to two antibodies simultaneously. The example include tumor marker detection. You can see here a comparison of each of these in terms of binding mechanism, sensitivity, speed, and the common applications. So direct CLIA is fast but less sensitive. Indirect CLIA increases sensitivity via signal amplification. Competitive CLIA is best for small molecule detection, and sandwich CLIA provides high specificity for larger molecules. Chemiluminescent labels are crucial in chemiluminescent immunoassays as they generate light upon chemical reaction, allowing for detection and quantification of the target analytes. Four common chemiluminescent labels are used in CLIA. These are luminol, acridinium esters, isoluminol, and enzyme-based signal amplification systems. So let's look into each one of these in detail. The mechanism of luminol. Luminol undergoes an oxidation reaction with hydrogen peroxide in presence of catalyst HRP, iron, or copper ions to emit blue light in the range of 425 nanometer. The reaction occurs in an alkaline medium and can be enhanced by enhancers like iodophene to increase the light intensity. The advantage is that it is highly sensitive due to strong luminescence, stable in dry formulation. The disadvantage is that the light emission is short-lived, requiring immediate detection and can be affected by interfering metal ions in the biological samples. This is used in Western blotting, CLIA for protein biomarkers, and forensic blood detection. Coming to the acridinium esters emit light when they undergo alkaline hydrolysis triggered by hydrogen peroxide in the basic solution. The oxidation reaction generates an excited state that releases energy as intense chemiluminescence. The wavelength range is 430 to 470 nanometer. The advantage is that there is no need for enzymes, making them faster and more stable than luminol based systems. A high quantum yield producing strong, long lasting signal. The disadvantage is that it can be hydrolyzed prematurely if exposed to high humidity and requires precise pH control for optimal performance. The applications include clinical immunoassays for hormone detection and infectious disease testing, and is also used in automated immunoassay analyzer for high throughput screening. Isoluminol is a structural analog of luminol with an additional amide group that increases the stability and solubility. Like luminol, it undergoes oxidation reaction with hydrogen peroxide, emitting light at a similar wavelength. The advantage is that it is more stable than luminol, especially in solution, and generates longer lasting signal for improving the detection efficiency. The disadvantage is that it requires a metal catalyst and less commonly used compared to the acridinium esters. Still, this has applications in high sensitive immunoassays for biomarkers in the clinical diagnostics. Next is the enzyme-based chemiluminescent systems. These systems uses enzymes to catalyze the chemiluminescent reaction for light emission. The two most common enzyme-based CLIA systems are HRP luminol system and the AMPPD system. The horseradish peroxidase catalyzes the oxidation of luminol in presence of the hydrogen peroxide, producing the light. The advantage is that it is well established in ELISA and CLIA platforms, and enhancers can boost the signal intensity. The disadvantage is that it can suffer from enzyme degradation over time. So stability is one of the concern. ALP-AMPPD system, this catalyzes the hydrolysis of AMPPD, triggering a chemiluminescent reaction. The advantage is that it generates longer lasting glow, making it ideal for high throughput automation. The major disadvantage is, however, it requires precise temperature control for the enzyme activity. Here you can see the comparison of different label types and their light emission wavelength, stability, etc. So in summary, luminol and isoluminol rely on oxidation reactions with hydrogen peroxide but need metal catalysts or HRP. Acridinium esters provide direct chemiluminescence without enzyme and are commonly used in clinical CLIA. The enzyme-based systems allow for enhanced signal generation and are widely used in the automated immunoassay platform. The chemiluminescent immunoassay offers several advantages over the traditional immunoassays like ELISA or calorimetric assays. CLIA uses chemiluminescent reaction rather than the calorimetric changes, allowing the detection at much lower unlight concentration. And the light intensity is directly proportional to the unlight concentration, enabling precise quantification. Acridinium esters and enzyme catalyzed reaction amplify the signal, making it CLIA more sensitive than the ELISA. For example, in cardiac biomarker testing like troponin 1 detection, CLIA can detect levels such as low picogram per milliliter, while ELISA may only be able to detect nanogram levels. It has wide dynamic range. CLIA covers a broad range of unlight concentration from very low to very high levels without requiring multiple sample dilutions. Unlike ELISA, where higher concentration may saturate the optical density reading, CLIA allows linear quantification across wider range. The example include CLIA-based hormone assays like TSH and cortisol can measure both the extremely low and high levels within a single assay run without significant modification. And CLIA provides faster reaction time. The chemiluminescent reactions occur instantly after triggering, allowing rapid detection. Many CLIA assays can use direct chemiluminescence, example acridinium ester based assays that do not require lengthy incubation steps like calorimetric assay. Enzyme enhanced CLIA provides strong signal within minutes. The automated chemiluminescent analyzers can process samples under 30 minutes, whereas ELISA takes 1 to 2 hours. CLIA also has a low background noise, improving signal to background noise. The CLIA eliminates the interference from sample turbidity, hemolysis, or color variation, which can affect the optical based assay. Like ELISA, chemiluminescent signals are generated only at the reaction site and do not require external light source, reducing noise from the background, fluorescence, or ambient light. Signal amplifiers and enzyme enhanced chemiluminescence further improve signal to noise ratio. So, due to these advantages, CLIA is widely used in clinical diagnostics, pharmaceuticals, and research, and it enables highly sensitive, rapid, precise detection of biomolecules, making it the gold standard for modern immunoassays. We have looked at the advantages of CLIA. Now let's see the limitations of CLIA. While chemiluminescent immunoassays offer superior sensitivity, rapid detection, and broad dynamic range, it also has certain limitations and that should be considered. It requires specialized instrumentation like luminometer. Unlike ELISA, which can be performed with basic spectrophotometer, CLIA requires a luminometer that detects the emitted light. And these instruments can be expensive and require specialized training for operation and maintenance. Some labels may degrade over time. Chemiluminescent labels such as acridinium esters and luminol derivatives can be sensitive to environmental factors such as temperature, light, and humidity. Enzyme-based labels such as HRP luminol system can lose activity over time. And there is a potential non-specific binding leading to false signals. The non-specific binding occurs when labels or antibodies bind to unintended targets, leading to false positive or false negative result. The chemiluminescent signal is highly sensitive, meaning even minor cross-reactivity can generate misleading data. While CLIA remains a more powerful tool, understanding and addressing these limitations will ensure more reliable and the reproducible results in clinical research applications. Now let's look at the applications of CLIA in much more detail. Chemiluminescent immunoassays is widely used in various fields due to high sensitivity, specificity, and rapid turnaround time. It is widely used in the clinical diagnostics. CLIA is one of the most commonly used technique in clinical laboratories for detecting biomarkers associated with diseases. It is used in hospitals, diagnostic centers, and research institutes. Let's look at some of the key examples. One is the cardiac markers like troponin 1 and troponin T used for early detection of myocardial infarction, that is the heart attack. Second is the BNP, brain natriuretic peptide, a marker for heart failure. Tumor markers such as PSA, CA125, and alpha fetoprotein, all are assays are based on the CLIA. Hormone assays such as TSH, cortisol, and insulin and C-peptides, all these are also detected using CLIA because CLIA provides higher sensitivity than ELISA, ensuring early detection and better patient monitoring. So it is also used in the infectious disease detection. It is for the pathogen detection, particularly in viral and bacterial infection, where early and accurate diagnosis is critical. It is used in HIV testing, hepatitis B and C testing, COVID-19 testing, and other infectious testing like dengue, Zika, and chikungunya. The rapid detection and high throughput testing make it very ideal for large-scale disease screening. So it is also used in pharmaceutical research in the biopharmaceutical industry for drug discovery, clinical trials, and biomarker validation. Due to its quantitative analysis nature and the high sensitivity, it is making it crucial for pharmaceutical research and clinical trials. It is also used in the food testing to ensure the safety of the safety and quality of the food. It's used in the food industry for detection of foodborne pathogens, detection of allergens and toxins, and contaminant screening. So here you can see the different applications and the examples of the analytes used, and why CLIA is important here in case of different testing purposes. Chemiluminescent immunoassays is a very powerful technique that has transformed the modern diagnostics. Its highly sensitive specificity and rapid turnaround time make it an ideal tool in the clinical, pharmaceutical, and food safety applications. So in this table, you can see the comparison of CLIA with our other immunoassay techniques like rapid immunoassay, radioimmunoassay, and ELISA. So based on the sensitivity, detection method, and equipment requirement, safety, and speed, I've populated these to have a comparison between different techniques for the detection. Let's look at the future trends in CLIA. Chemiluminescent immunoassays is continuously evolving with technological advancements to enhance sensitivity, specificity, and usability. Below is the detailed explanation of each of the emerging trend along with the practical applications. One is the multiplex CLIA for simultaneous detection of multiple analytes. So in this, it enables the simultaneous detection of multiple biomarkers in single reaction. Instead of performing multiple separate tests, one sample can be analyzed for different analytes. And this is achieved by using multiple chemiluminescent label that emit light at a different wavelength. So this is particularly useful for disease panel testing, personalized medicines, etc. It is time efficient and cost effective as it reduces the number of separate tests that is needed while maintaining the high accuracy. Second application is the integration of this technique with the microfluidic for point of care testing. The microfluidic layer combines miniaturized fluidic channels with chemiluminescence for rapid portable testing. The use of lab on a chip technology, which requires small sample volumes and integrates multiple tests into a single device. Mainly applications include bedside diagnostics, home-based testing devices, and the use of these in the resource-limited settings. The faster real-time diagnostics with the minimal equipment requirements is the key advantage of this technique. Third is the AI-driven data analysis for enhanced diagnostic accuracy. The artificial intelligence and machine learnings are used to analyze the CLIA results, and AI-based pattern recognition algorithms can distinguish between true signals and the background noise. This is useful in automated diagnosis and for error reduction and quality control, and also for clinical decision support systems. So this increases the accuracy, reproducibility, and the diagnostic confidence in reducing the risk of false diagnosis. Next is the nanoparticle based CLIA for ultra-sensitive detection. The use of nanoparticle like gold, silver, and quantum dots or the magnetic beads as a signal amplifiers. The nanoparticles enhances the luminescence signals, allowing for the detection of extremely low analyte concentration. Some examples include the early disease detection, ultra-sensitive infectious disease detection, environmental monitoring, etc. So these future advancements in CLIA are revolutionizing the diagnostics by making them more efficient, more accessible, more precise, and more sensitive. To summarize, CLIA offers high sensitivity and specificity, and it uses chemiluminescent label for detection, and it can be applied in clinical diagnostics, pharmaceuticals, and food safety. And future innovations include automation, AI integration, and advanced sensitivity improvements. CLIA will continue to evolving into more automated, faster, and ultra-sensitive diagnostic tools, making it indispensable for early disease detection and personalized medicine. That brings us to the end of this session on chemiluminescent immunoassay. I hope this deep dive have helped you understand its principles, applications, and future trends. If you found this video helpful, don't forget to like, share, and subscribe for more biotech content. See you in the next video. What aspect of CLIA do you find most interesting? Please comment below and subscribe for weekly biotech insights. Thank you.